Every living thing you have ever seen, touched, or been is built from proteins. The muscle pulling your hand toward this book, the enzyme breaking down your last meal, the antibody quietly patrolling your bloodstream right now, all of it comes down to protein, and protein comes down to shape. Here is the thing about shape. A protein starts life as a simple chain, a string of chemical units called amino acids, linked one after another like beads on a thread. That chain can be hundreds or even thousands of units long, and the moment it is released inside a cell, something almost miraculous happens. It folds. In a fraction of a second, that loose, floppy string collapses into a precise three-dimensional structure, a specific shape. The shape it was always meant to take, and that shape determines everything. Whether the protein carries oxygen, or cuts DNA, or builds a cell wall, or triggers a cascade of signals that tells your heart to keep beating. Scientists understood this much by the middle of the 20th century. What they could not do was predict it. Given the sequence of amino acids, given the beads on the thread, could you work out the final folded shape? In theory, yes. In practice, it was one of the hardest problems in all of biology. The number of possible ways a protein chain could fold is astronomically large. One estimate puts it higher than the number of atoms in the observable universe. Evolution had solved the puzzle over billions of years. Human science was stuck. For 50 years, researchers chipped away at it. They used X-ray crystallography, painstaking and slow, to photograph proteins one at a time. They built computational models. They argued at conferences. Progress came in fragments. The gap between knowing the sequence and knowing the shape remained stubbornly wide. Then, in 2020, a team from a London artificial intelligence company called DeepMind entered a biennial competition called CASP, the Critical Assessment of Protein Structure Prediction. They had entered before and done well. This time was different. This time, their system, AlphaFold, predicted protein shapes with an accuracy that matched experimental methods. Not approximately, not impressively close, matched. The scientists judging the competition used the word stunning. Some said it felt like cheating, before they accepted that it simply was not. What AlphaFold had learned, by training on decades of accumulated structural data, was something researchers had spent careers trying to articulate: the hidden grammar of folding, the deep logic connecting sequence to shape. A machine had cracked open a problem that had defined and in some ways defeated an entire generation of molecular biologists. The implications were immediate and vast. If you can predict shape, you can begin to understand function. And if you understand function, you can start to ask a far more powerful question. Can you design proteins that have never existed before? That question is where this story truly begins. Designing something that has never existed before requires more than knowing a protein's shape in isolation. It requires understanding the full molecular conversation, the way proteins grip onto strands of DNA, the way they cradle small drug molecules, the way RNA threads through their structures like a whispered instruction. AlphaFold 2 cracked open the door. AlphaFold 3 walked through it. The leap between the two versions is not merely incremental, it is architectural. Where AlphaFold 2 was built to handle proteins and proteins alone, AlphaFold 3 was redesigned from the ground up to model entire molecular complexes: proteins with DNA, proteins with RNA, proteins bound to the small organic molecules that pharmaceutical companies spend billions trying to develop. All of it together in a single unified system. To appreciate why this matters, consider how drugs actually work. Most of them function by latching onto a protein, sliding into a pocket, blocking a receptor, jamming a molecular switch. The interaction between a drug candidate and its target protein is the whole ballgame. For decades, Understanding that interaction meant expensive laboratory experiments, X-ray crystallography, months of painstaking work. AlphaFold 3 can now model that interaction computationally with a speed and accuracy that would have seemed like science fiction just a few years ago. The technical engine behind this upgrade is a class of artificial intelligence called a diffusion model. If you've heard of AI systems that generate images from text descriptions, you've encountered diffusion models in a different context. The principle is similar. The system learns to gradually refine a cloud of noise into a precise, structured output. In AlphaFold 3, that output is a three-dimensional molecular complex, atom by atom, resolved into something you can actually analyze and build upon. What makes this genuinely extraordinary is the scope of what can now be predicted. A researcher studying how a virus hijacks a human cell can model the viral protein alongside the human receptor it targets. A drug developer screening thousands of compounds can prioritize candidates based on how well they're predicted to fit their target. A geneticist studying how a transcription factor reads a stretch of DNA can watch that interaction play out in atomic detail without ever touching a test tube. Drug discovery pipelines around the world began shifting almost immediately after AlphaFold 3's release. Pharmaceutical companies that had spent years and fortunes on structural biology infrastructure suddenly had a computational tool that could compress timelines dramatically. Not replace the laboratory still matters enormously, but compress the early stages, cut the dead ends, and focus human effort where it counts most. The molecule is no longer a mystery to be solved one painstaking experiment at a time. It is a landscape that can now be mapped, explored, and ultimately reshaped. And that reshaping, the deliberate, intelligent design of molecules with specific functions, is the frontier this story is now approaching. Reshaping molecules, of course, is not an abstract pursuit. Behind every redesigned protein, every newly understood binding site, there is a patient, someone waiting for a treatment that does not yet exist, or one that exists but no longer works. Consider antibiotic resistance. It is one of the quietest catastrophes unfolding in modern medicine. Bacteria evolve, they mutate. They find ways to dismantle the drugs we throw at them, and the pipeline of new antibiotics has, for decades, moved far too slowly to keep pace. Developing a new antibiotic the traditional way takes years of laboratory work, failed trials, and enormous cost. AlphaFold 3 changes the math. By revealing the precise three-dimensional structure of bacterial proteins, the machinery that keeps those organisms alive, researchers can now identify vulnerabilities they could not see before. They can design molecules that fit those vulnerabilities like a key fits a lock. That is not a small thing. That is the difference between losing the race and having a fighting chance. Cancer research is feeling the same electricity. Tumors survive, in part, because the proteins driving their growth are difficult to target. Some of those proteins were once called undruggable, a word that carried a kind of grim finality. AlphaFold 3 is quietly retiring that word. When you can model how a cancer-driving protein folds, how it interacts with other molecules, and where it might be disrupted, the undruggable starts to look a great deal more targetable. The scientific community has taken notice, so has the financial one. Venture capital, which tends to move toward wherever the future is concentrating itself, has poured billions into AI-driven biology startups over the past few years. Pharmaceutical giants, never the fastest movers, have accelerated their own internal AI programs and begun acquiring smaller companies that built their entire identity around computational drug discovery. The logic is simple and urgent. The firm that learns to use these tools fastest will get to the clinic fastest. And getting to the clinic first, in this industry, is everything. What makes investors and scientists equally excited, which is itself a rare alignment, is that this is not hype built on a single promising result. The foundation is structural. The predictions are verifiable. The proteins are real. The data is accumulating. Every month, new research builds on what AlphaFold has made possible, and each new finding makes the next one easier to reach. There is a momentum here that feels different from previous cycles of biotechnology enthusiasm. It is grounded in something concrete, the ability to see, at atomic resolution, the machinery of life itself, and to begin, carefully and deliberately, intervening in it. The science is moving, the money is moving, and somewhere in a laboratory right now, a researcher is staring at a protein structure that, a decade ago, would have taken a career to determine. Today, it took seconds.